I'm going to, after the meeting, you can find this recording in the channel conversation on the Microsoft Stream. Okay, got it. All right, so welcome everybody. This is Dr. Granier. This is Bio 207, and today's goal is to quickly just hit the tissues. We're not going to spend a lot of time on epithelial and connective because you have those two lab projects that you're going to do. But I want to make sure we go over muscle and nervous system tissue because otherwise you're on your own with that. So this is your live session of me hitting that. Other piece of news, while we were waiting for the meeting to officially start, I took a poll of the two people and we decided that Tuesday, since it's Labor Day, I will set up a collaborate session on Tuesday from 8:15 to 9 to review the epithelial and connective tissue labs. So that way, there will be a session on Tuesday morning, and then we'll do our normal team session on Thursday morning for next week. Okay, so can you see my, can someone just like say yes? So do you see my slides? I still am sharing.
Yeah, I can see
Okay.
it.
All right. So again, in the last time we talked and in the activity on epithelial tissue, you're trying to figure out what is it that makes this tissue epithelium and not connective tissue. And the key thing to hone in on are there are these 5 characteristics, okay? And because many times these tissues are going to be barriers, we are going to want oh wait, cancel control. Sorry, whoever I'm giving control to. We are going to give these tissues, again, we want them to be effective. So we want the cells to be touching each other. We want the cells, again, because of what they're doing, sometimes they're a barrier, but they're also an opportunity to try to absorb or take in nutrients or take in substance. So we want the polarity of the membrane. So we want some of the proteins and some of the molecules, what did I do? Some of the materials on the top of the cell to then be a little bit different from the proteins and the embedded molecules on the plasma membrane on the bottom of the cell that are holding and maybe playing an anchoring role. Okay. So that is the polarity issue. We don't want any blood vessels. We don't want anything to push into the tissues because in order to let the blood vessels, again, point in, they would then have to break that barrier. And to break that barrier would mean that you are no longer an effective barrier. So that's part of the reason why we don't want the vessels. And when we have blood vessels, they are then going to come up and the capillaries are going to push really close to the lumen or the basement side of the cells, and we'll typically have an artery coming in and a vein going away, and the capillary here will be where the exchange of nutrients between the extracellular fluid and some of the cells living in this connective tissue underneath, as well as the cells of the basement layer of the epithelial tissue will get their nutrients. Okay. And so that basement membrane, that lamina, it's usually going to be a type of connective tissue of loose areolar to make room for our blood vessels to have space. It's going to be kind of an open, loosey-goosey type of connective tissue. All right. And these cells, there are going to be forces of friction as fluid, as air, as things are bombarding the tops. So we expect that in order to continuously replace the dead and dying cells, we are going to need to have a lot of mitosis or reproduction going on. Okay, go get ready for school. No, you can't have an iPad. Okay, mommy's working. All right, so for naming purposes, again, if it's single-celled, you would say that this epithelium is simple, and then the second part of the naming is what do the cells look like? So many of our barriers between the blood and the heart chambers, the blood in the capillaries, the blood in the arteries, the blood in the veins, they are lined with simple squamous, these pancake-like flattened cells that make a barrier, so the blood is technically encased inside the vessel, inside the heart chamber, and not oozing and spilling over into the extracellular areas. All right, so that's simple squamous. Simple cuboidal, we're going to see this in the nephron. We're going to see this in some other locations. Simple cuboidal and stratified cuboidal, we normally are going to see these two a lot in some of our glands. All right, simple columnar, we're going to see that a lot in the GI tract. And then pseudostratified columnar, we're going to see that in our tracheas and some of our upper airways. And then transitional is only in the bladder. And then stratified squamous, this is where we're going to have that protein keratin, all right? And your skin, your cutaneous membrane, it's going to be keratinized, stratified squamous, so there's a lot of keratin, but in the vagina, in the mouth, in the nose area, it's going to be non-keratinized because we want those areas to be less dry. We want them to be more moistened, more fluid-like, so there's less chance of break and tearing. So that'll be non-keratinized stratified squamous. Okay. Now, connective tissues are going to differ from epithelial tissues for lots of appearance reasons. And the other thing about the epithelial tissues is it's one cell repeated over and over and over again. Connective tissues can have many, many types of cells. All right. They can have fibrocytes, fibroblasts, adipocytes. They can have any of them immune cells, so lymphocytes, macrophages, neutrophils, monocytes, macrophage, all of those cells can be living in the tissue. Okay, and they're living in the tissue and they do not have to be touching. So cells can be somewhat an island unto themselves in a watery background material. And inside that watery background material, there will be some proteins. And the proteins of interest that we care about are going to be collagen, elastin, and reticular fibers, all right? And the reason why we care about those fibers is the presence, the amount, and the orientation of these fibers is going to influence potentially what we name the connective tissue, because strong, very organized, orderly collagen fibers, we're going to usually expect that kind of connective tissue to be our ligaments and our tendons. And so those are dense, regular connective tissues. A lot of elastin fibers that are going to, again, give a lot of bendability to things. Those are going to be in an organized pattern. Those are going to be our elastic tissues. And some of the ligaments in and around the vertebra are going to be full of that type of connective tissue. Reticular is going to be like your spleen, your liver, your lymph node areas, where I want to let it's like a big kind of tunnel, a big warehouse. I want to have a really open space, but I need to keep the ceiling and the floor from collapsing on each other as fluid moves into the system. So the reticular fiber is going to be that kind of fiber to give me an open warehouse, an open framework, so fluid and lots of cells can travel through with the blood or with the lymph. And my resident cells can anchor to those reticular fibers. And so as the fluid moves through, they don't have to necessarily have to go with the crowd. Okay. So when we especially talk about our standardized or our normal connective tissues, we expect the presence of these fibers. When we start getting into our specialized connective tissues, there might not be any of these fibers, as in the case of blood and lymph. There also might be different cells that you don't see anywhere else, and that's the chondrocytes for the cartilage, the osteocytes and osteoblasts and osteoclasts for bone. All right, and then red blood cells and platelets will be part of the formed elements of blood, and you should not see platelets and red blood cells anywhere else in any of the other tissues. If they're there, that might be considered inappropriate, that you had a break or tear in the blood at some point. Okay, so again, these are just a bunch of the cells that can show up in any of our connective tissues. And again, these guys, oh, sorry. these guys are all immune cells. Melanocytes, you're going to see where where color potentially can come into play. So in and around the basement layer of the skin, in and around some areas like the back of the eyeball, the retina. Macrophages are also, again, immune cells, and I'm making a hot mess with my pen. So, and then your mesenchymal cells are kind of like saying those are stem cells. And your fibroblast and fibrocytes, these are the cells that make your collagen, reticular, and elastic fibers. So in all of your connective tissue propers, if you have a collagen, a reticular, or an elastin fiber, then at some point there has to be a fibroblast or a fibrocyte in that tissue to produce that fiber. So as we get older, in our skin, these cells die and they're not replaced as easily. So what starts to lose its population? Collagen fibers in the dermis of our skin. And so what happens over time? Less collagen fibers, our skin is more saggy and we wrinkle and we start getting collagen implants. We start pushing collagen extraneously into our system, or we go and we find fat and we put fat to take the place of the collagen in our lips or in our skin or in our, wrinkly areas. Okay. All right. So that should be somewhat a good little overview to help you as you go through again those labs that we'll then review on Tuesday next week. And that should give you a good idea of, again, where to expect connective tissues, proper specialized connective tissues, and where to expect epithelial tissues, and what you're looking for when you're trying to, again, look at a slide that's probably has 4 or 5 different tissues in the picture, what to look for to try to differentiate, okay, where was the epithelial tissue, where's the connective tissue, and then probably to be able to see where the muscle is sitting. Okay. Now, an organ is going to be usually defined of when 2 or more tissues come together. Okay. Now, organs usually are going to have, again, some type of connective tissue capsule. Then they have some type of, again, some connective tissue that's underneath the capsule. Then they usually have some muscle. And then they usually have, again, if there's an opening in the middle, they'll have epithelial tissue lining there. Okay. Not every place do we have 2 or more tissues coming together forming a really distinctive organ. So in some situations, we have 2 tissues coming together. We usually have one type of epithelial tissue and one type of connective tissue, and they form an organ, but it's not a true organ in an organ system. It's just a membrane. Okay. And so these are 4 membranes that we can potentially have where they are made up of one type of connective tissue and one type of epithelial tissue traditionally, and they might be a part of a bigger organ, but they're kind of a little entity within that organ. So we want to give them a little bit more of a distinctive naming. Okay. So remember our serous membranes, that's where we have the visceral parietal pleural membrane, the visceral parietal pericardial membrane, the visceral parietal peritoneal membrane. They are usually made up of, I don't know why it keeps doing that. They are usually going to be made up of a simple squamous cell layer, okay? And then behind it will be underlying connective tissue. So there'll be fibroblasts, some collagen, and elastin fibers. Okay. All right. So that's our simple squamous layer and our underlying connective tissue. Okay. And then usually what happens is you put 2 of those serous membranes together and you form a cavity, and then these serous membranes release fluid into that area, and the fluid they release, again, remember capillaries are going to be in that underlying connective tissue. Some of that fluid from the capillaries is going to be taken into the cell, and then the cell will take some of those ions and nutrients and water molecules and create the serous fluid that ends up in the cavity. Okay, so that is our serous membranes. Okay, and we find those in our around the lungs, around the heart, and around the different organs of the GI tract. All right. The cutaneous membrane is going to be your skin. Okay. And so this is just another name for the epidermis is the keratinized stratified squamous layer, the underlying connective tissue, which is a little bit of a mix of immediately areolar connective tissue, dense irregular connective tissue, and then as you go deeper, some reticular tissue, all kind of blended together in, I guess, kind of like a I don't know. They're layers, but they kind of blend into each other. That is going to be known as the cutaneous membrane. And then when you get to chapter 5, you'll learn the hypodermis layer is the superficial fascia that keeps the cutaneous membrane or the skin connected to the underlying muscle tissue. And so another name for the hypodermis layer is known as the subcutaneous layer. Okay. And so what you as a nurse might find is that when you give people certain shots, if you use the really small needle to just penetrate past the skin, it's called a hypodermic or subcutaneous needle. And you give a shot sub-Q, you're trying to get the shot, whether it's insulin, whether it's some hormones, whether it's just the TB test or the flu vaccine, you're just trying to get everything into that layer known as the hypodermis, the subcutaneous layer that sits between the skin and the underlying muscle. Okay, so the cutaneous membrane, there's only one. That is your skin. That is your epidermis and dermis. And so another name for it as a membrane is the cutaneous membrane. All right, synovial membranes are going to be in synovial joints, and synovial joints are freely movable joints. They are incomplete membranes. So when I have my bone of my knee, the synovial membrane only kind of goes here and here. All right. It doesn't actually form a complete circle. It just kind of connects maybe the two areas. So between the two bones, I can have synovial fluid, and the bones help create with the membrane my joint capsule. Okay. Synovial membranes can also kind of create little bursa sacs. So that way they can, I'm trying to get rid of the other extraneous lines, but I'm not doing a very good job. So they can, again, help with, again, joint just having fluid there. So the areas that are in that joint capsule, like the cartilage, like some parts of the bone, they can help provide nutrients to those cells in those areas. But they also can have fluid there for shock absorption and lubrication. All right, so that leaves our mucous membranes. All right, and we will have a variety of mucous membranes. And mucous membranes, there can be simple columnar type, there can be simple cuboidal type. They are usually going to be, again, some type of epithelial tissue, but embedded with that epithelial tissue will be these goblet cells. And the goblet cells are going to secrete the fluid that is more robust than serous fluid, so it's called a mucousy fluid, and that mucousy fluid will sit on top of our simple columnar or our pseudostratified ciliated columnar. And again, for the airways, as the air comes in, little particles in the air will hopefully be trapped in that mucousy fluid and kept and purifying the air, kept in the upper airways, so the air only makes it down to the lungs. Okay, so again, it's kind of one of these things where they're not technically organs. Some of them are part of organs. And so they don't fit sometimes the best into another place to cover. So we talk about these 4 types of membranes now because they are in some ways some of the simplest of our organs because they are composed of one epithelial and one connective tissue. Okay. Questions? I'm trying to go back to you guys. Okay. Questions so far on any of this? Okay. All right. So hopefully, again, as you're reading, as you're working in your lab activities, that you will see how, again, all the epithelial tissues work, all the connective tissues work, and you'll get a little bit better handling of, okay, when I put one epithelial and one connective tissue together, I start to, again, form the simplest of organs. And in many cases, many of our organs are going to be two or three or four tissues come together, but we start that organ formation when we start to put one epithelial and one connective tissue together. Okay, back to my slides. So is everybody able to see these again? All right. So fascia, you won't see fascia mentioned anywhere else in the book, but again, fascia is the connective tissues that kind of bridge some of the gaps between the other organs. And so again, if the skin is our cutaneous membrane, and it's made up of the epidermis and the dermis, the skin as an entity, as an organ, has to then be connected to underlying tissue by some type of connective tissue. And that connective tissue is going to be your hypodermis. It's also going to be known as your superficial fascia. And so that is a very fat rich layer. It's also a reticular rich layer, and that's where usually your big arteries, your big veins, your big nerves are running. So when you are looking at someone and you see their blood, their veins, and their, they look blue, part of the reason why they look blue is because when light comes in and it hits the vein and then it bounces back, as it bounces back, some of the light energy is absorbed by the fat and dermis and the skin cells. And because they absorb the energy in the spectrum, what we then see with our eyes is blue, but it really isn't blue. So if you poke your arm, you're really not full of blue blood. Okay. Now, when you go and you get a massage, Swedish massages, the goal of a Swedish massage is to push enough pressure on the superficial fascia and a little bit of underlying muscle to try to get good blood flow to the body. Okay. When you go and you get a massage and you want to go with deeper pressure, now you're targeting the connective tissue that's between and around your smooth muscle that might be in and around the body cavity and your skeletal muscle that is in that region, and then the tendons and ligaments that are connecting the muscles to the bones. All right. And usually deeper pressure is required because you have to try to get, again, changes to the blood supply, changes to the fluid movements in layers of muscle that are below the skin, so the cutaneous membrane and the hypodermis and the subcutaneous membrane, right? And then the deepest of our connective tissues and our fascias are going to be that visceral fascia that is usually associated in the belly area with your peritoneal membranes. All right. And those, again, are also going to have, again, a mix of areolar connective tissue and adipocytes. And then when you talk about fat in the body, women tend to have more layers of subcutaneous fat because estrogen pushes us to store our fat in our subcutaneous or hypodermis layer. And so women tend to, again, distribute fat in that layer. Men, have you ever seen the beer guts of men? Their testosterone sometimes pushes fat to be distributed more in that belly and that gut area, that subserosa fascia, that not subcutaneous, that visceral fat, it's called. And again, it's the adipocytes in those connective tissues, usually associated with your visceral and peritoneal membranes. So all of this kind of, again, goes back to helping to connect. Again, the muscle distinctly is underneath and separate from the skin, and there's a layer of connective tissue between it. There's connective tissue in and around the muscle cells, so they're not touching each other. And then you have a distinctive kind of break between the muscle and then underlying organs with that subserosa connective tissue and fascia, right? Okay, so that's kind of how epithelial and tissues start to come together. And again, some of the importance of connective tissues that serves, they take up space and they fill in these gaps between our organs. So the organs are distinctive entities, but the space between them is not hollow. It's not always just like a fluid-filled sac. Okay, all right, so muscle tissues. All right, we have three types of muscle tissues in our body, and think of them as a spectrum. Skeletal muscle is on one side, and smooth muscle is going to be on the other, and cardiac is going to be in the center. Okay? No phones. Go get ready for your meetings. You have a meeting in 10 minutes. Sorry. Okay, so skeletal tissue is going to be, again, the organ system for our movement based, our heat production. It is going to be striated. It is going to be long cells, and the striations are going to make stripes. All right. And there are going to be lots of nuclei. So they are multinuclear, and they are going to have lots of mitochondria, and they are going to be wrapped in connective tissue, so that way the skeletal muscle cells do not touch each other. Okay. And the reason why we don't want them to touch each other is we don't want them to, when we ask a skeletal muscle to contract, we might only want a small number to contract. And so if they're touching each other, that would make the whole organ contract. So by keeping them as individuals, I can then say in my bicep muscle, I only want 20 of my skeletal muscle cells to contract. And if they're not touching anybody, they're wrapped in connective tissue, then I can use my nervous system to tell just those 15 to 20 cells, you guys contract and nobody else. Okay. Now, skeletal muscle cells, because they're multinucleated, they have a lot of mitochondria. It is not easy for these guys to go through mitosis, so they are going to be known as terminally differentiated cells. So right now, if I was to go and cut away part of your bicep muscle, those cells that I take out of your body, you're not going to get back unless you're really, really young. Okay? Those cells, the space that they end up being creating because you take them out, that space will get filled by, hopefully the cells that remain will take up the slack and have to hypertrophy and grow bigger in size. And then the connective tissues and the hypodermis and the deep fascia around the muscle will also kind of hypertrophy and take up that space. Okay, so one of the things about skeletal muscle is we don't see a lot of mitosis. We don't see a lot of, again, growth in terms of cell numbers when we exercise. We're normally working on making skeletal muscles increase in muscle mass by making the existing cells get bigger in size. Okay, all right, so if that's one end of the spectrum, then smooth muscles on the other. So smooth muscles are going to be these spherical cells, and they can touch each other. Right? They are only going to have one nucleus. They do not have stripes. And they can multiply and divide and increase in size and mass. And we see that especially when someone gets, let's say, they go into the heart and they take a vein and they graft the vein to, again, bypass an occlusion. The vein, which has a few muscle cells embedded in its layers around in the histological layers, the vein, with all of a sudden now having to act as an artery and having to deal with the high pressure of the fluid flowing through it, will actually hypertrophy its smooth muscle layers and hyperplasia, grow new smooth muscle cells from the existing one, and it'll slowly in a few weeks start to look like an artery because it'll start to have a nice thick artery layer of muscle that grows from the existing smooth muscle cells that were there in the beginning. Okay, so that's kind of the spectrum. Skeletal muscles, again, they're not touching. They are told to contract by a neuron. All right, and when they contract, they give you 100% effort. Smooth muscle cells can be told to contract by the neuron. They can be told to contract by local particles, local factors, local signaling molecules like adenosine, prostaglandin, different molecules like that can be released from immune cells. It can be released from the local cells in the area. They can also be told to contract by hormones. So the way I get a smooth muscle to contract is by a variety of mechanisms. The way I get a skeletal muscle to contract is by one way. The neuron tells it. When I ask a smooth muscle to contract, because they don't have the striations, they can contract by going, oh, I'll just contract a little bit. and I'll make my cells just a smidge, maybe 10% more contracted. And then I can ask them again with, again, maybe some more hormone, more nervous system innervation. I can say, nope, I need more contraction. And they can go, okay, I'll contract my cells 10% more. Okay, so they can give me, in a sense, graded contractions. They can give me a ramping into a more contracted state, or they can ramp out and give me a less contracted state. All right, skeletal muscle, they give you 100% or they give you 0. Okay, so that's kind of the spectrum. Cardiac fits in the center. Cardiac is going to be a distinctive cell with one nuclei, like smooth, but it's going to have striations. Cardiac is not going to be told to contract by the neuron, and it's not going to be told to contract by a lot of factors. Cardiac is going to have certain cells known as pacemaker cells that are going to be the ones to tell the rest of the cells to contract. Smooth muscle cells touch each other. Cardiac muscle cells touch each other, and in certain places, they will even merge their membranes and let the cytoplasm of one cell flow into the cytoplasm of the other cell. So they can be chemically as well as physically joined to each other. So when one nodal cell starts to tell everybody around it to contract, every cell in the cardiac that's connected will then start to contract because they're all chemically and physically linked. Okay. Cardiac cells are not going to be able to divide and multiply like skeletal muscle. And that, again, is one of the reasons why if you have a heart attack, if you have blocked arteries and cardiac cells start to die, you can't grow new cells in their place. And so eventually people who have lots of little occlusions and lots of little minor cardiac events slowly end up with a heart that has lots of dead areas. And those dead areas where the cardiomyocytes have gone away, they don't come back, they don't grow new cells in their place. What happens is some of the cardiac cells around might try to, like skeletal muscle, hypertrophy a little bit, but mostly what happens is the connective tissue that's in and around there, bringing the space for capillaries to come in and out, they'll just take up that space. And so an older person's heart who's gone through lots of little micro infarctions, lots of little bouts of occlusion and death to little minor spots in the heart, they actually, as an adult, have less cardiac cells and less contractile ability of their heart, and thus they start to go into what we call the heart failure condition. Okay. So those are kind of the distinctive tissues of muscle. So the big thing is they are normally going to be red in color, and the red color is going to be because they are going to have a lot of blood supply to them, and they need a lot of blood supply because they need a lot of oxygen to be able to make a lot of ATP. All right, so the red is going to come from blood being rich. The other reason they'll be red is inside the muscle cells, they are going to make this protein known as myoglobin. And myoglobin, like hemoglobin, has an iron core component, and iron can bind oxygen, and when iron binds oxygen, it turns red in color. And so that'll help also make the skeletal, cardiac, and smooth muscles also look very red in color. Okay, so when you're looking at your tissue samples, you're normally going to see, again, maybe in a tube, this very red and very cell-rich region that the cells might be arranged in a circular pattern, and then you'll see the cells kind of arranged longitudinally, so they'll kind of be coming in and out at you. Those are normally going to be our smooth muscle layers, all right? And then the cardiac tissue is only in the heart, and then skeletal muscle, you'll see it again running maybe in and around the outside of the tubes in very distinctive patterns, because their patterns is going to be to best then direct the force they're applying, so they can move the bones or move the body parts. Okay. All right. So again, here's just some of the some of the characteristics that I want you to know. So striated means those striped or band appearance, and we'll learn what makes the bands in cardiac and skeletal muscle later on. Non-striated, again, in smooth muscle, they still have some of the same proteins, they're just not arranged in the same manner, so they don't make that overlapping striped appearance. All right. Again, your cardiac and smooth are going to have one nucleus, whereas skeletal will be multiple. And part of this is why there is no mitosis, because it would be very hard to make a cell divide and equally distribute and equally in every single nuclei get the same pattern of DNA splitting when there's 20 nuclei to do that in. All right. Muscles can be controlled voluntarily. Again, this is part of the nervous system control of skeletal muscle. Okay, so skeletal muscle are only told to contract when the neuron tells them, and that neuron is going to be guided back to some areas of the brain that are going to, again, let you consciously think about, I want to make this contract, so I'm going to tell these muscles in this area of my body to contract. Smooth muscle and cardiac, again, because smooth muscle can be told in a variety of ways to contract, and when it contracts, it only gives you that 10% more or 20% more or less contraction. That is not under voluntary control. Like right now, I can't think, stomach, contract. That's not going to happen. Or smooth muscle in my small intestines, contract and make room because I want to go eat a big brunch. That's not going to happen. Okay. And in my heart, again, I can't tell my heart, contract more forcefully right now, or give me another heart contraction. All of that is under involuntary means. And part of that goes back to it's controlled in other mechanisms not directly tied to the nervous system and not directly tied to the areas of the brain where I have conscious control over that. Right? Other big thing is skeletal muscle and heart. They are either contracting at that 100% or 0% rate. So they give you an all or nothing response versus smooth muscle gives you 10% more contraction, 10% more contraction. And they can end up holding themselves in a state of contraction that might be the cell is 20 to 30% of its actin and myosin, its contractile proteins, or in a state of interaction and holding that. Okay. So that's kind of the thing about muscle. All right. One of the things with the heart and skeletal muscle is because they do not go through mitosis, because they don't multiply, the chances of having a cancer develop from a smooth skeletal muscle or a cardiac muscle cell is very, very low because they do not go through mitosis. Their cell cycle has them arrest and stop the process of mitosis. Smooth muscle could potentially, again, under the certain conditions, they're not known to be robust in cell mitosis, but they could give you more of a risk of a cancer developing. Okay. The implications of them not growing, not going through mitosis and multiplying in number means, again, if I take away some of my skeletal muscle or my cardiac tissue, I don't get it back. All right. And then there is an age where I could potentially still grow and multiply and replace lost cells, but that age is very much like in the uterus and at a very young toddler or underage. Okay. Here are some of the pictures. Again, for skeletal, you're looking for those stripes. You're looking for the multiple nuclei, and you're looking for the cell to be long and skinny. All right. Cardiac cell, you're looking for those breaks. You're looking for those areas where it's a band, but it's even thicker than normal. And the increased thickness comes from the proteins of the cell membrane on each membrane of the cells. The two cells are merging, creating even a thicker area of protein population, and then they're creating channels so cytoplasm can fit in between. The other thing about cardiac cells, you see how there's these forks, these spaces? They'll create space for, again, connective tissue to take up residence and for blood vessels to be able to push in between. All right, so they will connect to other cells in some locations, but not all, and they'll have part of their cell. It's not going to be a perfect square. It's not going to be a perfect rectangle. They'll have these forks and these breaks where they'll connect maybe to two different cells. Like in this case, this part could be connected to one cell, and this part could be connected to a different cell. And this guy is sharing cytoplasm with now two different cells over here, and maybe even two different cells over here. Okay. And then smooth muscle, you're looking for lack of striations. You're looking for a usually it's a densely populated area where, again, some of the cells will be arranged in a circle longitudinally. And when they contract, they make the whole of the circle, smaller. And then the long way, so if I have a tube going long, some of those smooth muscle cells are going to be lining up in a longitudinal fashion, and they'll make the tube, again, kind of collapse inward, so the length will go longer and then smaller. And that'll help, again, with mixing and mooshing and moving around my contents inside. All right. For nervous tissue right now, you're looking for tissue that is going to have a neuron and the supporting cells around it. All right. Neural tissue cells are going to be specialized to conduct electrical impulses. They are going to, again, have areas, usually one-way communication. So we typically draw them as cells. I think they look like little bushy heads like cyclopses, and they tend to have areas here with lots of membrane proteins that receive neurotransmitters. Then they have an area that has the voltage gated channels located, so they can electrically change the inside and outside of their ions so they can conduct an electrical signal. And then they have an exiting where they release neurotransmitter onto the next cell. All right. And around these cells will be supporters, and some of those supporters will be these, again, cells directly in contact and around the neuron. Others will be free floating in the extracellular space to contain anything that might try to get into the extracellular environment. All right. We see a lot of our nervous cells and supporting cells congregate in the brain and spinal cord. So that's why those are the two main organs. But coming off the brain and spinal cord are peripheral nerves. And so we'll see large numbers of pieces and parts of neurons. It might just be the axon, all right, that congregate together and work as a unit to then spread out and interact with the exterior parts of the body that are not the brain and the spinal cord. Okay, so for right now, whenever you think of neural tissue, you're looking for the functional cell, which is the neuron. So it's the cell that has created this little area right here, has all its voltage gated channels, and then this little area, and then this little area. And those voltage gated channels allow it to repeatedly produce a signal that is replicated over and over again in a manner that I can then transmit an electrical impulse down the neuron's axon. All right. This cell is, again, not a cell that you would easily be able to divide and multiply. So it is also, like skeletal muscle and cardiac tissue, a terminally differentiated cell. All right. And it is going to then not go through mitosis. So another place where we usually don't hear about people having cancers from the neurons, all right, the cancer usually is going to come from the supporting cells, the neuroglial cells. So sometimes you hear about people have brain cancers, but the brain cancer is actually a neuroblastoma, a glioblastoma, because it comes from the supporting cells, which do have mitosis and the ability to divide and multiply. All right. And for right now, all the supporting cells are going to be called neuroglial cells. We're not going to worry about their specific names. We'll do that at the end of the semester when we get to the neuron chapter. All right. So that is our nervous and our muscle tissue. Does anyone have any questions? Okay, let me see if I can go to my show conversations. Nope. Am I missing something? Did y'all? Okay, somebody did reply. Okay, so there are no questions. All right, so just to recap, I will try to get this recording posted either into Blackboard or on YouTube. You need to finish your epithelial and connective tissue labs. Hopefully this is helping you with the other lectures, the readings, to be prepared for that. And I am going to have a meeting and collaborate on Tuesday since we have the holiday. Okay, so I'm going to stop the recording, I think. All right. Hopefully that stopped. And if you don't have any questions, I will see you guys on Tuesday in Collaborate. All right. So have a good weekend, everybody. And hopefully this helps you get a little bit more through chapter 4. Tuesday, we'll go through the review of the epithelial connective tissues. Then next Thursday, it's our time to do skin. And then remember that you have a quiz coming up. The quiz will be open book, open notes. No respondus. It's just open book, open notes in Blackboard. And then we go week 4. We have our test, which is going to be over chapters 1, 4, and 5. And we'll have our lab over 1, 4, and 5. And those will require that you do the lockdown browser. Okay. So I hope everybody has a wonderful Labor Day. And I will see you Tuesday. And again, if you have any questions, feel free to email me. I'll try to be in Microsoft Teams tomorrow morning between 8:30 and 9:30. So if anyone does want to try to come in, I'll try to see, or just email me and I'll try to make sure there's a link if you want to try to ask questions or you want to go over anything. The tutor information, again, there should be tutors for the Merrimack campus through the academic support. And then I think I put the information for the Forest Park campus, the bone room. If you are struggling and you feel like I can't, she doesn't help me, and I've listened to Anatomy GMC, I just want someone else to try to help go over this with me, go and seek help from the tutors. Meg is one of the tutors at Merrimack, and she's awesome. All right, so utilize those sources, and guys, I will see you then on Tuesday. All right, I got to go. The alarms are going off. The kids are, of course, not at their meeting, so I'm going to go yell at my children. Okay, I'll see you guys.